Good afternoon, everyone. It is wonderful to see your faces here. We're really excited today because this is an inaugural named lecture for the Charles L. Cooney Lecture. And before I go into details and describe Charles Cooney and a little bit about his career, I just wanted to very briefly thank the sponsor of this lectureship, Nubar Fayan, who sponsored both the Daniel I.C. Wong Lecture and the Charles L. Cooney Lecture. He's our very own alumnus, and he entered the biotechnology area in its emergence as an academic field and in industry, completing his PhD in biochemical engineering here at MIT in 1987. During his career as inventor, entrepreneur, and CEO, he co-founded and helped build over 50 life science and technology startups, and these companies include Moderna, Kodiak, Denali, and many others. He's also written numerous scientific publications and is the inventor of more than 80 patents, and he is the founder of Flagship Ventures. He is also a senior lecturer, or has been a senior lecturer at MIT Sloan School of Management at Harvard's Business School as well. All right, it is a real pleasure to introduce this new lecture in our series. It's actually significant because it recognizes Charles Cooney. Charles Cooney is the Robert T. Haslam Professor Emeritus in the MIT Department of Chemical Engineering. He obtained his bachelor's degree in chemical engineering from the University of Pennsylvania and his master's degree and PhD in biochemical engineering from MIT. After working briefly at the Squibb Institute for Medical Research, he joined the faculty of MIT as an assistant professor in 1970. He's been a full professor in chemical engineering since 1982. He received the 1989 gold medal of the Institute of Biotechnological Studies in London, the Food, Pharmaceutical, and Bioengineering Award from the AICHE, and the James Van Lannen Distinguished Service Award from the American Chemical Society's Division of Microbial and Biochemical Technology. He is also a founding fellow of the American Institute of Biological and Medical Engineers, as well as a fellow of the American Chemical Society in 2009. He is a consultant to multiple biotech and pharma companies, and he has sat on the boards of companies such as Genzyme, Polypore International, and Biocon Limited. His research and teaching span many aspects of biochemical engineering and pharmaceutical manufacturing. He holds more than 30 patents, has published over 250 research papers, and has co-authored or edited 5 books, including Development of Sustainable Bioprocesses, Modeling and Assessment. And in addition to his professional interests, he is a trustee of the Boston Ballet, an overseer of the Boston Symphony Orchestra, and a board member of MIT's Community Service Fund. His other interests have included rock climbing, skiing, high-altitude mountaineering, to which he has used for many examples in discussing entrepreneurship. And he's also done ascents in Denali, Amadablam, Mont Blanc, Kilimanjaro, and Huascarán, scuba diving, and antique map collecting. So he is an incredibly multifaceted individual. I can speak to my own experience with Charlie. I was a UROP in his lab, where I learned some of my first bioseparations processes, and also a student in his classroom, where he inspired me. He continues to inspire large numbers of our undergraduates. He teaches a very important part of our undergraduate biochemical engineering. It's really a core senior class that is taught in the spring, in which students learn about how to build a biomanufacturing process. But they learn a lot more than that. He shows them how to integrate everything from the economics to some of the social and ethical impacts of the work. He has also been important to the world of entrepreneurship here at MIT. He has served as the director of the Deshpande Center for Entrepreneurship. He has held that role for many years and actually helped to establish the Deshpande Center. And in that way, he launched many other careers. So in many ways, Charlie has launched the careers of students and alumni and many others beyond MIT in the biotechnology arena. It's for that reason that this inaugural lecture is really dedicated to the new foundational leaders in biotechnology, people who stand at the frontier of new areas in bioprocess and biotechnology. And our first inaugural speaker is Dr. Armon Sharei. He is the Chief Executive Officer and founder of SQZ Biotechnologies, a clinical stage biotechnology company focused on unlocking the full potential of cell therapies for patients around the world. It has active programs in oncology, autoimmune, and infectious diseases, and other serious conditions. Dr. Sharei received his bachelor's in chemical engineering with honors and distinction from Stanford University. He earned his PhD in chemical engineering right here at MIT in laboratories of Professors Bob Langer and Klaus Jensen. During that time, he created and implemented the cell squeeze technology as a novel method for rapid and flexible cell engineering. Prior to joining SQZ as CEO, and just show a few pictures of him here. I love this one with the squeezed cell, very happily engaging with Armand. Prior to joining SQZ Biotech as CEO, Dr. Sharei completed a Reagan Institute postdoctoral fellowship at Harvard Medical School in the laboratory of Ulrich von Adrian. Dr. Sharei has published 19 peer-reviewed publications and is the inventor on 20 patent families. We're very excited to hear him talk today, so I'm going to turn things over to Armand.
Thank you very much, Paula, and thanks a lot to the department and Nubar and Charlie for having me. It's definitely an honor to be here after having been a student here. I realized almost 10 years ago, which makes me feel very old. But definitely been an exciting journey. And my hope with the talk was to kind of do a mix of the science and kind of business entrepreneurship side to hopefully keep everyone interested enough in what our journey was. And it's definitely an interesting experience being in the room where I was sitting there listening to a lot of classes, being in the thorn of the side of some of the collaborators in the room and my PIs. So apologies in advance for all the things I did back then. So just for some quick background, as far as where we are right now, to help give context to the backstory, we started SQZ back in 2013, around when I was graduating. Right now, we're a public company. We're about 130 people. Throughout the journey, we've raised about $300 million in equity financing, and then we have a large partnership with Roche, which is over $1 billion. And the core of the technology is something almost ridiculously simple. We just discovered that if you squish cells at high speeds, you can disrupt the membrane and put whatever you want inside them. And this has been the basis for our therapeutic programs that are in the clinic right now. Now, to give some context on the backstory going into MIT, I was born in the US, but I grew up in Iran and Dubai, and I did my undergrad at Stanford, and I actually really wanted to go straight into industry. That's us visiting a uranium mine during my senior year. I didn't want to go into uranium mines, but it was a cool shot. We were doing masks before it was cool. And I really wanted to go to the industry side, but my professor at the time, Jinan Bao, was saying, don't be an idiot, go to grad school. I'm like, fine. So I came here and I started working on a project that was in Klaus's lab and then got co-advised with Bob and Klaus, which was a high throughput microinjection system, which I'll get to in a minute. And just to get things going at the time, I was a mix of very hardworking and determined, but also lazy. So I got a lot of undergrads to help. This is a picture of the first cohort of UROPS. I think by the time I left, I had like 15 or 18. Klaus and Bob maybe knew about half of them, but they were getting concerned by the numbers. But they were great. They were all happy, and they're all doing good things now. And the one in the middle in the purple actually did most of the cartoons you guys see. So what is the story of kind of how SQZ came about? So the background to how we think about the field, because in the end, our whole goal was to try to really enable engineering of cells, especially as it relates to being able to generate cell therapies. And if you think about the world of therapeutics, you have small molecules, which were probably arguably the beginning of modern medicine, and then you've had biologics, where people can do more sophisticated engineering and manipulation of the biology in the body. And then the newer wave is things like cell therapies, where you can now engineer a cell, which is ultimately a molecular machine that can do many different things. And if you think about what it takes to make these new waves of modalities come about, is that what it took to go from small molecules to biologics wasn't that suddenly someone thought injecting proteins is a great idea. People were doing that for a long time. There was pig insulin, there was placental factor VIII. What really changed everything was recombinant DNA technology was invented, and now you could suddenly make whatever biologic you wanted, and you could implement it at scale. And now you have this world of biologics that have made a huge difference for many patients. Cell therapies are in a similar spot where right now people can make cell therapies, but it's very complicated to do it. It's very expensive. There's a lot of constraints on what you can do with these cells. But if you could solve that problem, you can unleash a whole new world of therapeutics that could potentially do far more even than what the biologics and small molecules have done. Now, if you think about what it takes to make a cell therapy, it's pretty different from the others. So small molecules are all chemistry. Biologics are all fermentation. Cell therapies, your two ingredients are what's the cell, what's the material you're going to put inside it, and then that's going to give you your engineered cell that is your therapeutic. And if you think about what are the limiting ingredients here, it might be a little counterintuitive. So the cells themselves are actually not that limiting. These cells either exist or they don't. You're taking them from your body, you're taking them from a donor's body. Making synthetic cells is not that much of a thing yet, at least as far as I know. And the materials are really not that limiting either, because the field has done a lot of work inventing all kinds of molecular machines that can do things inside of cells. There's siRNA, mRNA, DNA, CRISPR, all these great things that you can use to manipulate a cell. But what's been difficult is really putting these two ingredients together. That has constrained what kind of functions you can engineer within cells. Can you do it in a scalable way? Is the cell normal on the other end? And that's where the field has really had two major constraints. One is the biological constraints. So because of what cell types you can deal with, what materials you can deal with, how functional these cells are after they've been treated, it's really limited what therapeutic applications you can use a cell therapy for. The second part of cell therapies, which often gets more airtime, is the manufacturing problem. So it's very expensive, it's very time-consuming to do this. Is it ever really going to be a sustainable solution to diseases? So to try to address this problem, there was an Italian postdoc in Klaus's lab that had a very American solution, which was, let's shoot the cells. So this gun system, the way it worked was you had a pressurized chamber, you had cells going by, and you would shoot at them at high speeds as they went by to teach them a lesson and put the material you want inside them. And this stainless steel chamber to the right is kind of to give you a feel of what this really looked like in real life. Now, as with many grad school projects, you sign up to one thing, it turns out to be a different thing, and that life wasn't as simple as that cartoon implied. It wasn't really working. And so we were trying to solve this problem, and one thing we thought was an issue is as you would shoot at these cells, cells are pretty plastic, so they'll just deflect away. So you would shoot at them with the gun, but they wouldn't actually get penetrated necessarily. They would just deflect away and zip on by. So we thought, okay, let's really jam those cells up against the nozzle so they can't deflect as much, and now when you shoot at them, it should go through. So we had set up a system that looked something like this, and this is a high-speed camera image, but basically you would have It doesn't want to work. You would have cells zipping through the channel at high speeds, and that gun element you see is the thing that would try to fire at the cell. There we go. Is the thing that would try to fire at the cell as it passed by. Now, we had started to get this to work after having squished the cell up against the gun enough, but then the parameters made no sense. So whether the gun was set to high or low, it didn't seem to matter. So one day we just turned off the gun, and it turned out the whole thing still worked. We're like, great. So basically it turned out just the squishing step was enough to disrupt the membrane and get whatever you wanted inside. And so it seemed the core of what we had discovered is simply take a cell, mix it with whatever you're trying to deliver, run it through this constriction. It's going to disrupt the membrane temporarily, and then anything that's outside is going to diffuse in. Cells will detect that disruption and close it back up. And what was interesting is that this principle seemed to be very broadly applicable. So this worked across every mammalian cell type that we tried, be it various immune cells, stem cells, red blood cells, whatever you're thinking of. And because it's just a diffusion-mediated delivery, it didn't really matter what was outside that you were trying to deliver. If it was soluble, it would diffuse into the cell. The other piece of this was somewhat counterintuitively, the cells were surprisingly happy on the other end of this. So these smiley faces aren't fake. The cells seemed totally fine. Like if you looked at their gene expression or looked at their behaviors afterwards as far as proliferation or differentiation or other functions, they seemed to be fine. And what we thought might be the case here is this is just happening because there's ultimately highly conserved membrane repair mechanisms in cells. And these mechanisms are designed to just detect the disruption, close it up, and not think much of it. So collectively, now that you've had all this flexibility on cell type and cargo, as well as the cell health and function, now it looks like you can basically engineer any biology you want and start to implement it for many different disease areas. The other side of this was because it's such a simple concept, it really lent itself to scaling. So by putting many channels in parallel, you could make this really high throughput. So that picture you see there is of the clinical version of the chip. The line you see down the middle right here, that has about 1,000 channels in parallel in it. And that's what lets you run these chips at about 10 billion cells a minute. So it runs super fast. If it's red blood cells, you're in the trillions per minute category. So the simplicity of the technology really lent it to scaling. And what was really different from the existing systems people were using, as depicted in these disparaging cartoons, is right now in the cell therapy field, people either use viruses or electroporation. So with viruses, you basically engineered a virus to try to enter the cell and put in the cargo. With electroporation, you're essentially electrocuting the cells to try to drive cargo inside them. And on different metrics like cell health, scalability, diversity of cell type and cargo, et cetera, it looked like with Squeeze you could overcome a lot of the problems that we were having in the field and therefore could open up a lot of new areas. So kind of going to the what do you do about this side, just from a timing perspective, I started my PhD in 2008. We invented this around 2010, and then fast forward a few years, we were collaborating with a lot of different groups, both within MIT and outside of it. It looks like there was something exciting going on, and we were starting to debate, do we start a company? And then I was like, okay, this could be interesting, and I was talking to Bob and Klaus was like, well, I was like, how do we do this? Like, who should run it? He's like, well, you can just run it. I'm like, no, Klaus, you're crazy. I can't do that. So after those debates, we started the company, but I didn't run it. I was very involved, but I didn't run it. And the main debate we really had when we were setting Squeeze up was, do we want to be a therapeutic or do we want to be a tool? In the sense that Squeeze as a tool can do many things. You can go down a path of making this tool available to many people and helping them develop new technologies. Or you can go down the path of let's make our own cell therapies and take that all the way into patients. So I always really liked the therapeutic side more, but the tool side seemed very accessible at the time. So we started the company off of $100K from Bob and Klaus and my parents. My parents had set aside $30K for a first mortgage. They said, are you sure you want to waste that money on this psychotic idea of yours? I'm like, yes. So, because it's going to be a great use of that money. So we got the company going, and I actually went and did a postdoc in parallel at Harvard Med School in the immunology side because it looked like the immunology side of what we could do was really interesting, and can we make exciting therapeutics out of this? So we started the company in 2013. About a year and a half in, this is what our life looked like. This is meant to reflect that it was very messy. You don't need to follow any of the lines or the people, but it was meant to reflect that our life had gotten very, very messy in the sense that we had a lot of funding sources on the academic side from various grants and internal groups, but then we also had the company side, which had its own angel funding. It had its little piggy bank of what we had started off with. We were making some money off of selling the chips. And then there was this messy network of people that were in the company versus were in the academic side, or were in the academic side but controlling people on the company side. There was a lot going on. And it was getting unsustainable as far as living in both worlds, at least for me. So what started to really precipitate a change was I met through Bob, our now chair, Amy, who had just left Pfizer. She was kind of big shot like pharma, biotech side person, and we had met her through Bob, and she had gotten very interested in this, and she was kind of pushing the question of like, why don't you just switch to the company side? Like, why don't you get the company to do the therapeutic stuff? And I was very confused because I was like, I really want to go down the academic route. This sounds fun. When you're on the academic side, you kind of view the other side as the dark side. I'm like, I don't want to be on the dark side. So there was a lot of debates, and I was talking to my professors and advisors a lot. And one memorable piece was I went and was doing my whining to Bob about I can't decide. And then Bob says, he probably remembers it, he's like, follow your heart. And then I'm like, really, Mr. Barry, one of the most published engineers of all time, you're telling me something as non-quantitative as follow your heart? But he was right. I was way overthinking it, and in the end, there was no good answer. And it's just like, just pick and stop complaining. And part of it was, if you think this is the most exciting thing you'll ever do, you should probably jump. If you think you'll come up with other things down the line, don't jump. Wait for those other ones. And I was like, no, I'm pretty sure this can be the biggest thing ever, so that's why I jumped. Not that I'm not biased. So I switched to the company side, and at that point, the problem we were really trying to solve was the problem of cancer vaccines. So to give you context, in the world of cancer, there's pretty clear evidence that killer T cells, or your CD8 T cells, have a huge role to play in destroying the tumor. So for example, checkpoint inhibitor drugs, which are ubiquitous at this point, have this correlation where if a patient responds to a checkpoint inhibitor, they have a correlated increase in CD8 tumor infiltration. If a patient does not respond to a checkpoint inhibitor, you won't see a correlated increase. And this is true across many different tumor types. So there's been many analyses across all kinds of tumor types. You can't really get, or there's a clear relationship between CD8 T cell responses and ultimate outcome for the patient. And if you think about the world of vaccines, you normally think of them as a prophylactic only. Like, you don't think about, oh, I have this disease, I'm going to take a vaccine for it. It's either you got it beforehand and it saved you, or if you already have it, vaccine is not your answer. And that comes down to the fact that most vaccines are very good at CD4 responses and antibody responses. That's why, I don't know, when you think about COVID vaccines, you're hearing about antibody titers. You're not really hearing about CD8 counts. But what you really need for a therapeutic intervention is these killer T cells. And there's been many attempts in the cancer vaccine space. By and large, they have not worked out, and the only one that's ever been approved is primarily a CD4 and antibody mechanism. It's not a CD8 mechanism. So this is a problem that we really wanted to solve. And at the core of the problem, if you think about how T cell activation works, it comes down to antigen presentation. And the way antigen presentation ultimately works for most vaccines is that you have an antigen that gets endocytosed by the antigen-presenting cell. When it gets endocytosed, it's primarily going to get processed through the MHC class II pathway, which presents the CD4 T cells and can subsequently support antibody responses. Very little ever makes it to the MHC class I pathway. Whereas with Squeeze, what you can do is load the antigens directly into the cytoplasm, and that is where MHC class I primarily samples from. So now you can shortcut the system and get direct class I presentation. So earlier this year, our team published in JI on how simply by changing the location of the antigen, where it's in the cytosol instead of in the endosome, you could get this massive improvement in CD8 T cell activation. And so we thought this was really promising as far as now you can potentially solve that big problem of a cancer vaccine that would actually work. And the project we were starting to work on and is now in trials was to take immune cells out of the blood, squeeze them with the target antigen for the tumor, and then once they're presenting the antigen, simply inject them back in. And the idea was that these cells should then home to the appropriate lymphoid organs, talk to the CD8 T cells, induce their activation, and then have them go and kill the tumor for you. So this is the project that we were working on as our kind of lead program. And it was a bit of an unpopular thing to do, because at the time, this was in 2015, there was a lot of excitement about cell therapies, but it was about a different zone of cell therapies. It was all about CAR T treatment. So CAR Ts had just started to show really dramatic effects in certain hem-onc indications. There were a lot of companies coming out around it. It was a really exciting time for it, but we were doing an atypical thing of saying, yeah, we could go down the CAR T route, but we think this cancer vaccine problem can be a much more exciting one to solve, even though it wasn't necessarily trendy at the time, because there had been a lot of failures in cancer vaccines. And luckily for us, Roche also got really excited about what we were doing, so even though we were in a pretty nascent stage, they came in with a large collaboration, which really helped put a lot of wind in our sails, because now it was like, oh, okay, an adult thinks this is a good idea. Otherwise, like, what are these people thinking going and doing cancer vaccines when everybody's doing CAR Ts? So this was at the end of 2015 when Roche did the partnership. As a result of that, we started to grow. We started to recruit more professional-looking people than us. And we also got our first VC funding around that same time. And then we faced an interesting choice where we have this Roche collaboration going, and our choice is, do we really focus on the Roche collaboration, try to drive that forward, because with the money from Roche, you could have sustained it for quite a while and just really gotten that going, or do we take a chance and expand further, go after some of the more exotic other ideas we had and see if they can turn into something, because now you're parlaying that Roche validation into something bigger, and can you use that to gain more momentum? And as we think about kind of what kind of difference Squeeze was making for the field, there's two dimensions to it. One of them is for existing mechanisms that people are pursuing, we could have made them, or we can make them, much better from a purely time and cost perspective, and to an extent, quality. So what I mean is right now, for example, there's a lot of T-cell therapies that people develop. And with these T-cell therapies in production, there's always a proliferation step, or almost always a proliferation step. And with their standard practice of using things like electroporation for editing these cells, there's actually a significant deficiency in how well the cells proliferate. But with Squeeze, they proliferate as normal because the function is normal. So that meant that you could get to dose faster. And in a world, and that sounds trivial, but in a world where time of production in the clean room is a massive cost driver, this meant that if you could produce in half the time, you were probably going to produce with half the costs. And for cell therapies, where cost is a massive barrier, this could be a real efficiency for these companies. The other side of this is cell function. So for hematopoietic stem cell-based treatments, there's issues with how well the cells can engraft or how well they can differentiate after they've been treated with something like electroporation, which meant that people need to put long hold times in their manufacturing. But if you could get rid of that hold time because your cells were totally normal, now you could also, again, cut down on time and cost. The other side was the simplicity of Squeeze meant that it was easier to integrate it into a broader machine that could do the end-to-end production. So now if you have end-to-end production that can deal with the wash steps and bag fill, et cetera, you can have a closed system that won't need a clean room anymore. And at this point, you've also automated it. You need fewer operators to run it. And that can even further change what the cost basis is, but also what the accessibility is. Because if you can actually make these cell therapies at the hospital site instead of at a central manufacturing location, that would really expand access and change the cost dynamics of making cell therapies practical. The second part of this was, can you do new things biologically? So one way to think about the new things was, can you take current concepts and make them much better from a safety and efficacy standpoint? One example, going back to T-cell manipulation, is that right now for things like TCR therapies or TIL therapies, so TIL therapies, for example, you're taking T-cells out of a patient's tumor, expanding them out, and then putting them back in so that they'll go kill the tumor. One thing they need to do is, because of these expansions, these T-cells become really exhausted and their phenotype is not great. So you need to administer IL-2 into these patients, which drives a lot of toxicity. But with Squeeze, we showed that you could use mRNAs to express membrane-bound cytokines. And with these membrane-bound cytokines, now the T-cell basically has the signals it needs itself. It doesn't need the systemic administration. And what it means is your T-cells should be perfectly functional, if not even more powerful, but you're not causing broad toxicity from doing it. And this may sound trivial for us that we're at least used to mouse stuff, but humans complain when you inject them with IL-2. Mice don't. And they have good reason to complain because it's very toxic and it hospitalizes them. So by doing this, you can really change the safety profile and potentially the efficacy of these treatments and make them something that's much more broadly applicable across tumor types, but also can be applied earlier in the treatment setting. The other side was, can you create new therapeutics that weren't even possible? So the vaccine mechanisms I mentioned, these are things that are really hard to do with current techniques. The other side is regenerative medicine, where because we can deliver transcription factors into cells, we can help direct their differentiation into different cell types, and by doing that, overcome some of the long-standing issues in Regen Med, where it's been really hard to direct cell fate. But if you can load a transcription factor, you can start to direct cell fate in a much more effective way. So these are the kinds of areas, especially category 2, where we started to expand in. We were trying to think about, like, what else can we do? What other concepts can we go after? So throughout that time, we were growing more and more. The team was getting larger. We were looking more and more professional in pictures. And also the amount of cash that we were burning was going up too. In 2018, we did yet another big raise. Roche came and doubled down on our collaboration together because they had really liked our first program, but then we had a second program that they didn't have rights to that they were really intrigued by. And it was a complementary mechanism to what we were doing with them. So that resulted in them doubling down on the collaboration. And things were really moving, but our life was also getting a lot more expensive. And for us, kind of in a blink of an eye, we had gone from spending a few million a year to many millions a year. And the next big milestone was going to be showing clinical data that this actually matters. And you face an interesting dilemma when you get to these scales, because you're increasingly in a zone where private financing is more and more difficult, because they're not set up to put these large amounts of money later and later. So your option is, can you just go public? Or do you wait on data and then go public? And it's kind of an interesting trade-off, because if you already have good data, it's easier to go public. But what if you don't have good data? Then you're screwed. So a lot of people face this debate of, do you go before or after you have your data? And there's a lot of pros and cons to thinking about going public. Generally, there's that deeper pool of capital. The financing mechanisms are a little bit simpler and more streamlined, and you have access to a much broader type of investor mindsets. Like, some of them might be short-termists, like they only want the 3-month flip. Others might be multi-year, but they can all have very different time horizons. Whereas VCs, by nature of their setup, when you start, that's time 0 for them, and then they kind of need to be out at some point. They can't stay there forever. Now, the cons are life is more complicated when you're public. There's a lot more compliance stuff. There's a lot more overhead for G&A things. The environment can be more unforgiving. Like, every day you have a share price, whereas when you're private, no one really knows what you're worth. It all exists on paper in some mysterious way. But there's a lot of, there's always a spotlight when you're public. And also, people can do short selling when you're public. So if you're about a private company and you think their idea is garbage, there's nothing you can do about it except say you think they suck. If they're public, you can go and short sell them, which means that there's much more intensity on people betting for and against things, whereas on the private side, you can only really bet for something, not against it. The final consideration, which I think weighs heavily on a lot of people, is if you try to go public but fail, it is a very, for lack of a better word, public disaster. So it can be really problematic if you try and you can't pull it off. So despite all of the hand-wringing on the topic, we decided to go for it. And one thing in particular that made it more complicated for us is that the way the system works, for lack of a better term, is that we weren't, I would call it a trust fund baby, in the sense that Squeeze was very bootstrapped. We were never birthed with a massive round, because those things make life a lot easier and more streamlined. The hedge funds, et cetera, that come in when you're public, they're used to that setup. They can get behind it because they know there's real momentum there. When you were this bootstrapped, strange thing, it's a little harder to get them to go behind it. So luckily, we made it out there, and one very exciting moment was seeing the New York Stock Exchange all lit up with Squeeze logos, which is pretty cool. So that was fun, and they had this all day, which was crazy. This is during COVID, so there's far fewer people on the floor. There's some people on the floor, but it was pretty cool to see that. And since then, thanks to that piece, we were able to continue going with all of our programs. At this point in our trials, the way it works for patients is that they get leukapheresis, meaning that you're taking their white blood cells, and then it gets shipped to our manufacturing site. In under 24 hours, we're able to process all of their cells, and then it's about 6 to 7 days to QC that. So in the end of the day, the patient is waiting about a week to get their material back. And the nice thing is the patients don't need to get preconditioned. They don't need to get hospitalized after this. And last year, I think our most exciting moment yet was to see from our last data update that we had a patient who was actually responding. So the whole point of this was to try to go out there and make a difference, and it's definitely a touching moment to see you actually manage to help someone. And right now, we're continuing to build on that momentum, both by making the manufacturing side even simpler and more streamlined, where with the point-of-care version of the machine, you can really condense this down to not need a clean room and be more readily available at any hospital site, but also using our flexibility of engineering to engineer even more sophistication into these cells. So the newer generation APCs, what we're doing, which is in trial now, is building on that theme of antigen presentation, we switched to mRNA-based cargos. So thanks to people like Nubar and Moderna and all these other companies, mRNA has become much more ubiquitous and easy to deal with, so supply was a lot easier. And now we went for a system where you can deliver 5 different mRNAs at once, where 2 of these mRNAs code for the antigen, so E6 and E7 are the 2 antigens you want to use for HPV-driven tumors. But then the other mRNAs code for really powerful stimulatory domains like CD86 and membrane-bound IL-2 and 12. And the interesting consideration here is that any one of these, what you would call signal 2 and signal 3 domains, are really toxic if you do them systemically. Things like IL-2, IL-12, CD86's counterpart is CD28, which had an infamous trial. These are really dangerous if you did them systemically, but if you've localized them just to your APC of interest, now you're really only giving the T cells you want that big kick, but the rest of the system doesn't really see anything. So these graphs kind of give you a sense where if you just had the stimulatory domains, not much happens. But if you have the antigen and the stimulatory domains, you can really crank up the intensity. So the idea behind this was leveraging Squeeze's abilities to further optimize what these cell therapies are and how powerful they can be. So later this year, we'll know more about this trial, but we're really excited to see what it can do. Beyond that, we've been working on other cell engineering concepts. So aside from engineering antigen presentation through APCs, there's been another thing we've been doing with red blood cells, where in this case, what we do is we use Squeeze to load red blood cells with antigen and adjuvant, and we make them appear aged. Now, the interesting thing with aged red blood cells is that they get cleared by professional APCs that are in your lymphoid organs. And these professional APCs are the best of the best at talking to T cells. The problem is they're really hard to get at. They don't circulate in the blood. They're hard to recreate ex vivo. It's a little mysterious exactly how they work their black magic. But in our case, it kind of doesn't matter because our red blood cells are going to just act as carriers to get the material to them. So these red blood cells are cleared within about 20 minutes. And what we saw in mouse systems is that we could get massive CD8 responses in the tumor. These responses are highly specific against the antigen. And not only can you kill the tumor, but you can also form memory from it. So it's doing all the things you would want it to do. So now in this trial, similar to the other trial I was talking about, we're looking at it for last-line patients that have HPV-driven tumors. And then from there, we would look at combining it with earlier lines and also combining it with things like chemo, which is in the earlier settings, because it appears to be very synergistic with chemotherapeutic drugs as well. And as an aside, that's one of the exciting things about cancer vaccine mechanisms, if you can get them to work, is that they should be very clean from a safety profile, because generally when you think of vaccines, you're not really worried about any kind of safety issue. So it really lends itself to being used in an early setting for these patients, or even going on top of other regimens, because you're not going to add to the toxicity issues. The other trial that we have that's going to be our next trial coming up is using a similar mechanism, but actually doing the inverse, so shutting down immune responses in an antigen-specific way. So it's the same concept. The difference is if you don't have an adjuvant in the red blood cell, the default outcome of this destruction is actually immune tolerance. So it's like the body constantly reminds itself to never attack an RBC. If you have an adjuvant in there, you're flipping the system to activating, but if you don't have an adjuvant, the system will remain tolerogenic, and this allows you to shut down immune responses in a target-specific way. And this is kind of the holy grail for autoimmune diseases, because right now for autoimmune disease treatments, all these drugs are basically broad immune suppressants. It's very hard for them to hit the piece of the immune response that's causing trouble. It just suppresses everything. But in this case, we showed that you can delete autoreactive T cells, you can induce antigen-specific Tregs, and these antigen-specific Tregs are capable of bystander suppression, meaning that they will shut down other T cells in that zone, even if they have different specificity. And we've done a lot of this work in type 1 diabetes systems, where we had a paper earlier this year where we showed if you load the red blood cells with the correct antigen, you can stop the onset of diabetes in these mice. But if you load them with the incorrect antigen, you don't change anything. Like, the mice will still develop diabetes, really illustrating that it's not a broadly suppressive effect, it's an antigen-specific effect. And for this trial, we're starting with celiac disease, because there are certain nuances about how you can do celiac disease trials versus type 1 diabetes trials, where in celiac disease, one unique feature is you can do what you would call a gluten challenge. So basically, you treat the patient, and then you tell them to go eat some bread and see what happens. And the hope is if it worked, they won't have a negative reaction to the gluten. So as we've been continuing the therapeutic side, in the background, we've been trying to do more and more to improve the manufacturing, where I had mentioned with the current manufacturing system, we've already been showing just how much faster and simpler you can make production of these cell therapies, where for us, it's been on average 17 hours, whereas normally it's days to produce a cell therapy. And kind of going back to my comments on clean room time, like, I know in some ways it may intuitively feel like, who cares if you had to wait a few days? But the cost is very linear with time in the clean room. So if something takes 10 times as long to make, it costs 10 times as much. And most cell therapies are on the order of over $100,000 just to make them. That's not what they're priced at. It's just to make them. It's that expensive, which can be unsustainable for a lot of areas. And that's where moving to these point-of-care machines that can be out of the clean room can be really exciting, where in the long run, we hope that you can actually do the production of these cell therapies at the clinical site and bring it closer to the patient and make it that much more accessible. So to kind of start zooming it back out, on the pipeline side for us, our 3 active trials are in the oncology side, so the APC and EAPC programs I mentioned. The APCs is the one that's part of the Roche partnership, and that's the one that had that exciting patient response end of last year, and we actually got FDA Fast Track designation earlier this year for that. So we're excited to see what these programs can do, especially given that the whole goal of starting Squeeze and jumping to the dark side was to try to make a difference for people. And the long-term vision we're trying to work towards is that if you think about cell therapies today, they tend to be a very expensive multi-week production process from a patient perspective. They are a far from pleasant experience given the toxicities associated with the preconditioning and the treatment itself. And they're actually pretty limited in where they can work. They work for certain areas, but they struggle outside of that. And with our current trial, we think we're already starting to show just how different a cell therapy can be, because our production is so much faster and more cost-effective. From a patient perspective, it's already much friendlier because they don't need to get hospitalized, they don't need to get preconditioned. And the biologies we're pursuing, if we can really get it to work, can be really broadly applicable. And we think this sets up for a future where you can actually do manufacturing on site, have 24-hour turnarounds for these patients, and make it very broadly accessible such that cell therapies can really be a more ubiquitous treatment, not something that's confined to major centers in the rich world. So kind of looking at the trajectory of Squeeze and the journey we've been through so far, there were a lot of different moments along the way as far as getting the company started and our initial collaborations, our initial funding, eventually IPOing. But I think the really important dimension I wanted to emphasize is how important the people component was throughout this, because none of those moments would have happened without the people that were involved with it, in the sense that from the very early days, if it weren't for all of your ops who helped get so much work done, way beyond what any one person could have done, we wouldn't have nearly made enough progress to break out like this. People like Klaus and Bob and all the collaborators we had back in the day made a huge difference in us learning about these different zones, refining our ideas, figuring out what to do with it, getting external people like Amy involved who helped to teach me the ropes about being not a scientist, or not just being a scientist, and the team that it takes to get all this done, but also the partners. So things like the Roche partnership wouldn't have happened without people like Christian, who used to be the head of oncology at Roche PRAD and is now the head of R&D at Bayer, like people like him who would take a chance on, or people like him or our investors that would take a chance on such an early concept and be like, okay, there's 50,000 reasons I can see you guys failing, but there's a chance. And if it works, it'll be great. So without them, you really couldn't get here. And also the patients themselves, the people that are sadly looking for solutions to what is otherwise an unsolvable problem for them, and then they're willing to take a chance on, okay, these crazy kids at MIT came up with something, sure, like, what other option do I have? And really taking a chance on doing these trials, because that's also for them and their families not an easy thing, how to deal with these decisions when they're in an unfortunate situation. So a few thoughts before going to the questions, because it's more entrepreneurship focused. I'll probably say starting a company is not for everyone. I know from the outside we make it seem like unicorns and rainbows all the way. It is not that. There's a lot of pain and suffering along the way, so careful what you're signing up for. I think in the end, I'm happy with what we did, but I would say it's because I love the impact component. I just like the science. And I say that in contrast to if I loved the science and just liked the impact, I don't think you would be happy doing what I do. Because there's all kinds of things you deal with along the way. But if you really care about making the difference, it makes you power through those pieces that come up as you go. And like I was emphasizing in that last slide, and I know this sounds like a cliche you hear all the time, but the people part is the most important part. They're the ones that help make that difference in the end. You walk into it thinking, if I'm smart enough, if I have the right science, any numbskull can be around me and we'll make this work. It's not the case. So that component shouldn't be underestimated. And in the long run, our hope is to live up to the dream that if we can really solve the problem of how do you tell a cell what to do, and do it in a scalable way, can we really underpin a new universe of cell therapies that can ultimately make a really big difference for these patients that we're trying to serve. So with that, thanks again, and happy to talk through any questions. Oh boy.
So, Armand, you gave the perfect inaugural lecture for the Charles L. Cooney Lectureship. And we really thank you for bringing your insights, sharing your experience, and giving us a sense of what it's like to have that kind of real-life impact. So thank you very much, and I'd like to present
Thank you very much.
the lectureship.
Thanks a lot. Oh, you want to share?
And with this, we're going to open up questions to the audience, and I see a hand right now, so we're going to get a microphone right up to Arun.
Hi, I have a question on the first part, the T-cell part. So one of the things that there's some data for, I don't know how strongly you can believe it, but I think it's not just noise,
Yeah.
that the mRNA vaccines give you a lower CD8 T cell response than, say, the adenovirus vaccines.
Yeah.
And the speculation there is that that's because it's a viral vector, so it gives you an inflammatory environment, which then leads to co-stimulation through CD80 and CD86. So I'm just curious, since you beautifully stick that already into the APC, have you ever compared giving antigens in a mouse model through your method versus natural infection with the same bug and see if you get the same level? Because then you've really got a strong response.
Yeah, so we have not, and one frustrating technical challenge with mRNAs is that for some reason, unstimulated mouse T cells and B cells and stuff don't express mRNA that well, whereas human cells express it great. Activated T cells in mouse express it just fine. And it's strange because delivery is not a problem. Like we can deliver to unstimulated mouse cells without an issue. They just seem to be more quiescent. So it's a long way of saying we haven't been able to compare in a syngeneic mouse system what this does. We've done humanized mouse stuff, but that's varying levels of artificial. But unfortunately, we've never been able to do that head-to-head in a mouse system because something about mRNA in unstimulated mouse cells doesn't work that well.
And one minor comment on the second part, in autoimmunity, which was very exciting too. But there's a recent finding that Tregs are not the ones that are doing the real expansion during. So there's another class of suppressor CD8s, and so it may be something to worry about in that space.
No, that's good to know.
Yes, we have a question over here.
If you just shout it.
You can shout it out.
Thank you very much for your talk. And I have a question, technical question. So correct me if I'm wrong. To me, the rate limiting step, like for cell therapy, is the in vitro cell expression. So I see your technology could speed up, put molecule into the cells. But how you speed up, the in vitro cell expansion or something? You mentioned you shrink from multi-weeks to 1 week.
Yeah. So it all comes down to what is the mechanism of the cell therapy, because some cell therapies outside of squeeze don't have expansions, like HSC editing. They're not expanding anything. They just rest the cells after electroporation. You're right that if your biology requires proliferation, using squeeze for that doesn't inherently change that. One thing that is different is, if you look at this part, the proliferation rate can be very different. So let's say you edited your cells using electroporation and then you're expanding them. If they've been electroporated, they tend to have kind of like a dip and then eventually catch up. And that really prolongs your time. So if certain snapshots, in this case at 7 days, you would have 16x the dose with squeeze. And it's purely because the electroporated cells are deficient in their expansion. So that helps cut down the time. For all these cases, it comes to the nuance of what is that cell therapy. And different ones, to your point, will have different rate limiting steps. So some are solvable, some might be beyond what squeeze would solve.
Armand, thank you. What are the constraints or the rules about what you can load into a cell? And how does that vary with the type of the cell or properties of the cell itself?
So it gets interesting to think about it. In general, if you can deliver one thing into a cell type, you can almost deliver anything because squeeze is fundamentally just disrupting the membrane. The rest is diffusion. So the kinds of cargoes you can load into any particular cell range from impermeable small molecules to mRNA, DNA, et cetera. We've done nanoparticles. We've done whole virus. Once you get to things that are above 100 nanometers, it starts to get dicey. We think that starts to become the limit of the pore size from what we've done. But basically, if you can get it in solution, and if it's under 100 nanometers, I'm pretty sure it'll go in. Across different cell types, you change things like the dimensions of the constriction, like how wide it is. So normally, this graphic doesn't totally scale, but Normally, it's about half the diameter of the cell is the constriction dimension. That's the rough guideline. So slightly larger cells would require slightly larger constrictions, et cetera. Speed matters, how fast you send the cells through. Those are the two most important variables. You can play with stuff like temperature. Temperature, we think, primarily affects the membrane recovery time. So it actually allows you to load more if you have it cold, which is interesting. In general, it's worked for every mammalian cell type we've tried. The ones that were trickiest were RBCs, because you had to go quite tight for that one, because they're used to getting squished all the time. And the membrane repair dynamics for RBCs are different, because nucleated cells have active membrane repair mechanisms. They will patch up the membrane, and they all more or less act the same, whether it's like a T cell or a stem cell. Red blood cells are totally different, because it's a thermodynamic membrane rearrangement. There is no active mechanism, so they thermodynamically reseal. It's a little bit of a different dynamic how they work. Something with a cell wall would be problematic. We briefly tried that. Not surprisingly, cell walls just clog up the whole thing.
Hey, thanks for the great talk. I was really inspired to meet you today because I'm also very interested in APC therapies.
Oh, cool.
But I have some entrepreneurship-related questions for you, and that was, what was your patent and IP strategy like in being able to protect not just the device, but also the cell products and expanding into different types of cells as well?
Yeah. So one lucky thing for us was Squeeze was a totally unexpected find. Like, no one had ever thought this was a good idea, ourselves included. Like, it was an accident. If you told us go squish cells at high speed, we'd be like, okay, what are you going to do? So on that front, we were pretty lucky in being able to file through the MIT licensing office. And at this point, like, the patents are granted globally. At this point, I would say there's kind of three buckets for the IP. There's the fundamental platform stuff, which is like the fundamentals of squeezing. There's the machines that run squeezing. So whether it's the point of care machine or the GMP machine. And then the third and broadest category is the what do you do with it component, like what are all the therapeutics you can create. I would say in our formative days, it was all about the platform stuff. We had some ideas on the therapeutic stuff, and there were some filings on that, but they were less mature than they are now. But that was enough to get going. I think my guess is as long as you have a good enough core that says tomorrow that other person can't just immediately copy you and get away with it, that's as good as it's going to get, and that's enough to get going, because your life will be determined by way more than the IP distinction beyond that. I think the CRISPR companies are a good example. Like, they have overlapping stuff, and that doesn't really hold them back. There's all kinds of other things that cause trouble for them, not that.
So I have a question, like you're involved the obviously the red blood cell, squeeze the red blood cell. So I wonder, like for like sickle cells, which are not like a normal
Yep.
blood, which are not normal red blood cells. So how would that affect?
We haven't tried sickle cells, so I don't know what that would do. In general, I think as long as there's no cell wall, it should probably work, because the speed at which this occurs is super fast. So in the end, it's not about did the cell decide to deform. It's like really you jammed it through a constriction, and no matter what its shape otherwise was, this phenomenon happens. I say that because adherent cell types like cancer lines or fibroblasts, they're not perfectly spherical once they're in suspension. They're all kinds of weird shapes. And that works fine, because at these speeds and under these tensions, it doesn't matter what you're doing. So I would expect those to be okay. But we've never done it.
Great talk. And I have a question about the manufacturing side. So squeeze itself happens pretty fast, but then you have a slower recovery before you go to the QC, which QC can also happen on the chip. So in the future, for integrated system, do you think it's a better approach to integrate all of them on the same chip and then letting the recovery happening on the same device, or actually after squeezing go directly into bulk? That's a better strategy.
The QC stuff in a weird way, I think comes down a lot more to what the regulators will be fine with as opposed to too much of a technical issue. Because in the end, they have certain things they care about. And for different cell therapies, it can be different. And how they care about it, if that makes sense, is what can be rate limiting. So what I mean is, one of the longest steps is sterility. And there's a compendial sterility test, meaning like the standard sterility test, that's 2 weeks. And we had to do a lot to make the case to use this machine that was otherwise standard in other zones to do a 2-day sterility test. And that was a whole thing to do that. So it wasn't even anything innovative. It was purely, hey, this machine is okay for other biologics and it takes 2 days. Why don't you let us use that for this instead of the 2-week thing? And that was already a major accomplishment. So a lot of times, I don't think that'll come down too much to a technical issue. For the point of care system, it's possible that if you've really made reliable QC metrics that fit onto an integrated machine, you might be able to get them to accept that. But those things need to be ridiculously robust. And it can be a journey for every single QC test for them to be okay with it. Right now, I think with the point of care machine, if you can just get rid of sterility, and that would be purely the argument of it's a closed system, it was patient material on, patient material off. If the system wasn't opened at any point, you should assume it's sterile. That alone, I think, would change a lot about the turnaround time. Because stuff like flow-based assays on viability and other things, those are fast.
Thanks, Armand. I'll ask the last question because I know we're near the end. First of all, we really appreciate your talking about all of these transitions and these decisions that you made along this path. And I'm wondering about the decision on disease area, because oncology is an excellent area, but certainly a lot of competition. It ended up being a winner, but how did you end up deciding on oncology?
So I would say jury is still out on whether or not we made the right choice, because if the vaccine stuff works, we'll look like geniuses. If it doesn't work, they'll be like, well, we could have told you because cancer vaccines always fail. So I think the main balance we faced at the time was, I think picking oncology was relatively easy because cell therapies were accepted for oncology, but if you did it outside, there would be way too many questions and anxieties about it. Because oncology is typically where people are most willing to be adventurous with new therapies. Because unfortunately for the patients, like this is their last shot. They don't care. They're in a sad way. They're like, just give me something. So I think oncology in that way is an easy choice. But what you do within it, we had an interesting balance of, do we go the CAR T route and show that we could be version, 2.5, that's that much better than the other ones, or do we go do something that no one could have done and like leapfrog the zone? And a lot of people would have picked option 1, but we picked option 2 because our mindset was, we can do things others can't. Let's put our money where our mouth is. And again, if we're right in the end, it'll seem brilliant that we did it that way. But if we're wrong, it would have been a very predictable self-inflicted wound in their view.
Excellent. Well, thank you, Armand, for this excellent lecture.
Thank you.